So now it's the time for testing this hypothesis. So what they have done, they take this LuxX gene and they transcribe this gene, they make a LuxX protein. So you have a LuxS, so made a LuxS protein. This protein is added into a test tube. So in a test tube we add SRH, then add some LuxS protein, and after some time of incubation, what we have seen this products, homocysteine and dihydroxypentanedione, or DPD. Okay, so this is a very moment, this is a moment of clap, something is discovered, is really interesting, really fascinating, something which is not known for many years is finally discovered after some time, that this part of the gene is nothing but, so let me erase all this. This part of the gene is nothing but LuxS. So this is the protein which helps to convert the SRH into homocysteine and DPD molecule. Okay, so after cracking the code of the protein, finally now we have to discover the structure of this LuxS as well as the DPD structure. So to go into that, we need to go through different sets of portions. And to discover the structure, the scientist encounters another major problem. What is the problem? Normally what they are looking at when the whole system is going on in vitro, these homocysteine and DPDs are made, but after the making of this homocysteine and DPD molecules, what happens is SRH, which is the ribose homocysteine residue. This is fairly unstable, not actually unstable, fairly very active. This molecule is really very active molecule, very active molecule. This is very active molecule that they can be converted into some other molecules. So all the time the ribose ring, in this case what we are looking at, so this is the pentameric ring of ribose sugar, as we all know. This ring is attached to this molecule, and this ring is really unstable and very reactive, that all the time the rings is broken, and this breaking of the ring and the joining of the ring. So the ring and the linear form can be formed throughout the time, and the mechanism is so that the equilibrium is so dynamic, the equilibrium is so fast, that there's a problem for scientists to actually understand this whole structure of this SRH when it presents in this situation. After many years of research, active research, finally the scientists from different regions, from different parts of universities, the crystallographer scientists crack the structure of this molecule. They're finally able to crystallize this molecule, and finally after the crystallization of the molecule you end up with the structure something different. So this is the structure of this SRH molecule, the molecule. Here is the ribose ring and is the homocysteine residues out there. Okay, so in this case this is converted by LuxS into DPD and homocysteine. So these are these. So normally the DPD is present in this form. In many times the DPD can be formed in other forms. So there is an equilibrium. So this equilibrium is so rapid that it's very difficult to snap the structure of a protein via different techniques that are available of the molecular structurization. Okay. So after many years of research, after many times of research, finally crystallize the structure of this molecule. And finally, when we crystallize the structure of the molecule, we make something like that. Okay. So here is the LuxP. Remember what is LuxP? So let's go backward down our slide. Here is the LuxP. So LuxP is the carrier molecule. Remember the carrier molecule, because we need a carrier molecule which will bind to the autoinducer 2 to carry the autoinducer 2 from the outer space throughout the periplasmic space, and finally attach it with the receptor. So LuxP molecule presents in the periplasmic space of those bacteria. And what is the structure of the LuxP? So normally after many years we have a chance to actually crystallize the structure of LuxP along with that autoinducer molecule which is presented in the middle here. So this is the autoinducer molecule you can see in the middle. So it's like a pocket in the middle. So there is a small or tiny pocket in the middle of this LuxP protein. So LuxP protein is made of a different several alpha helices. Because it's a transmembrane, it's a periplasmic protein. Always it has to be present in the membrane. That's why it is made up with different alpha helices. And in the core of this molecule, there are loop-like structure. In the core of this loop structure, we have this autoinducer molecule. So this is the autoinducer molecule 2, because as we know, in case of only the autoinducer 2 molecule, you need to have the carrier molecule, which is LuxP in this case. Okay, so now let's zoom on the structure. If we zoom on to the structure, finally, and here, this is the different residues, different cysteine residues of LuxP, which will interact with this autoinducer molecule. What is this autoinducer molecule? If we look at the autoinducer, which is the DPD, in this case, this is made up with the different rings. Two rings are there, two pentameric rings are there, and these rings, are red colored molecules, are the oxygens, and these are the methyl groups out there. And this black colored molecule are the methyl groups out there. Okay, and here is another moiety over there. What is this moiety? For the first time, normally people think about is the carbon. But if we think about the carbon, the whole system cannot be done, or the bonding cannot be explained. For after some time, we finally finally came to know that this part of the region is not made up with carbon. This is in fact another molecule which is very, very abundant in the marine water, is boron. Okay, so the presence of boron again thumbs up for our hypothesis, for the hypothesis of scientists, that this kind of system, this kind of auto-inducing factors is present in the marine water, because Vibrio fischeri and Vibrio harvey, both types of bacteria are found vigorously in the marine water. And in the marine water, there are lots of boron atoms out there, and those boron is engaged to make this ring. And finally, you have boron, and boron have a tendency to bind with the hydroxyl residue. So there are different hydroxyl residues. It will bind with the hydrogen bonding throughout this place to maintain the structure of this autoinducer molecule in the pocket made by the LuxP protein. So this is electron density structure of this LuxP hollow enzyme. And in the middle here we have the autoinducer molecule, which is much clearly viewed in this case. So here it is the formation strategy of autoinducer molecules. So what we have this DPD molecule here. So DPD is in turn is not act as an autoinducer molecule. It is further folded, because as I have told before, that this DPD molecule is very, very reactive. It will fold, it will just open its structure and fold and open, and all this time throughout this place. So it folds to make a DHMF structure like that, and finally it refolds again with the presence of water. The hydrolysis is done and the bonds are made and break, and finally the borate salt is there, and the presence of this borate salt, the boron atom, adds and to make a final ring, to make the second pentameric ring, and it will attach to the hydroxyl residues here, hydroxyl molecules there. The one thing I recommend you to notice in this picture is that the orientation of hydroxyl and methyl groups. In all this case, the hydroxyl group present in the upward and methyl is in the horizontal line. So this is the presence. So the hydroxyl here, methyl here. Okay. So now if we look for different sets of molecules, not AI2, AI1 pathway. If we look at the AI1 pathway, we can find the orientation will be different. The orientation will be methyl and hydrogen and hydroxyl like that. Okay, when we will discuss the picture, we will discuss it again. Okay, so these are the folding steps are going through. The first thing, it will need water to make the cyclization. After making the cyclization, the dehydration takes place, and as well as the dehydration, it will have to have the borate salt. Finally, to attach the boron into the top here, the top position here, and finally makes the THMF borate molecule, which acts as the autoinducer 2. So this is one type of autoinducer 2 that is designed. There are lots of different varieties of autoinducer 2 molecules can be found here, but in most of the cases in the marine waters, we find boron in this place as autoinducer, make autoinducer molecule. So here we have an operon system. That operon system now describes us how this autoinducer 2 system can be placed together along with the autoinducer 1 system, because in a real time, the autoinducer 2 and autoinducer 1, both system is carried out in the same place. So you need to have different sets of genes. Those genes, in case of Salmonella and E. coli, this whole thing is studied in detail, and you can see different sets of genes are out there to interact or counteract with all this kind of system, because Al2 molecules can be present in many different types of bacterial strains, but AI1 or autoinducer 1 is present in very few bacteria, because this is very specific. So in the AI1 system or autoinducer, AI2 system or autoinducer 2 system molecule, in case of Salmonella, they have a genetic sequence like that. There are huge genetic sequence, but we just take some of those genes, which we'll discuss in detail in this picture. So there is a C, D, B. So these are the 4 genes that are very important in this case to carry on the signal. We have seen the autoinducer 2 pathway in case of Vibrio harvey. Now let's see for the Salmonella. They are really relatively similar, as we can see here. So here it is. This is the product of gene LSRB, and it will make the molecule which binds with this pentameric ring. As this pentameric ring suggests us that what is this? This is the autoinducer molecule, autoinducer 2 molecule. So again, remember the presence of LuxP molecule or LuxP protein in case of Vibrio harvey, which acts as the carrier of AI2. In this case of Salmonella, it is LSRB, which carries the autoinducer 2 and carries it into the receptor. Again, in this case, this is a transmembrane receptor. It made up with 2 different genes via 2 different subunits of proteins, LSRC and LSRD. So this LSRC and D is present there, and LSRB along with the autoinducer 2 molecule attaches with these subunits, and finally it triggers the conformational change. After some conformational change, it attaches with the LSRA subunit, which is the blockage here, which is the blockage region here, which blocks the entry of simple molecules into the inside the cell, which present in the cytoplasmic portion of the cell. Here is the LSRA is attached to the LSRC and D. And whenever the LSRB along with autoinducer 2 binds with the LSRC and B, a conformational change is occurred, and then the LSRA, which is the ATPase or ATPase molecule, it harvests the ATP or energy of ATP, hydrolyzes the ATP into ADP plus inorganic phosphate, and finally it drives the entry of this autoinducer molecule inside the cell. But remember one thing again in this case, look very carefully, the LSRB molecule is restricted to be entered into the cell, because it is the huge molecule to be entered into the cell. So the LSRB remains there. Only after the conformational shift occurs and the ATP hydrolysis occurs, only the autoinducer 2 molecule enters the cell. After the entrance of the autoinducer 2 molecule into the cell, it will bind to different protein molecules there, and finally DNA binding molecule, and it will transcribe, and after the transcription, translation is occurred and the protein is made to carry on virulence or making biofilms and all this sequence of sequential structures. Here we have a core or hollow structure of LSRB molecule. In case of LSRP, we have seen the structure of LSRP, and you can see these 2 types of structure. They are really very similar structures. In this case too, we are looking at. So let me go backward. Here we have the structure of LuxP, and now let's talk about the structure of here, which is the LSRB. The LuxP molecule, both, and LSRB molecule, the structure are similar in many ways. That it is also made up with the alpha helices, and some beta sheets are there in this case. And in this case, the pocket is slightly large, but still the basic idea is the same. It is making a core, and in the core region there is a small pocket, and this small pocket, we can see here in the small pocket, the autoinducer 2 molecule can bind. After the binding of autoinducer 2, conformation changes, and finally it will bind with the LSRC and D. So there are different regions to interact with LSRCD molecules to finally make this autoinducer 2 molecule to be entered. Okay. So in all the different strains of bacteria that have been studied till now, we can find in case of autoinducer 2 system, the carrier molecule structure is more or less the same in all these cases. And as well as the autoinducer molecule structure can also be having a typical characteristics. But what is the basic difference between the autoinducer in case of Salmonella and the autoinducer 2 in case of Vibrio harvey is designated here. So this is the autoinducer molecule of Vibrio harvey. This is for Salmonella typhimurium. But we can look at this. This is a 2 pentamer, and this is not 2. This is only one ring in this case. But the basic difference is, in case of harvey, there is a presence of boron atom here. But in case of Salmonella, there is no boron, because as we know, Salmonella normally loves to stay in our gut, and in our gut there is no boron atoms there. So there are lots of different atoms, but not the borons. That's why they are lacking the boron atom in their in a place of their ring structure. And another difference, what I have designated earlier, is that here is the oxygen is, and the hydroxyl is here, and the methyl group is here. But in this case, what we are looking at, methyl is in the top, and hydroxyl is in the horizontal line. So this is another difference. And most of the bacteria which are studied, maybe their structure are different, but these are the things that are conserved. In case of in case of those marine bacteria, we can find this boron, and also the orientation of methyl and hydroxyl group are different than the Salmonella and E. coli. Okay, so these are the system of how the auto-inducing system 1 and the auto-inducing system 2 can interact with each other in both of the time. For example, here we have bacteria, sufficient amount of bacteria. For example, let me draw some bacteria. Here are the red bacteria out there. So, and let me change the color. And for example, yes, there are blue bacteria out there. So there are these two types of bacteria: blue bacteria and red bacteria. Blue bacteria can talk with blue bacteria via auto-inducing 1. So they can interact via AI1 machinery. Red can interact with the red via auto-inducer 1 machinery. But if the red bacteria want to talk or communicate with blue bacteria, they need auto-inducer 2 machinery. And in the same case, if the blue bacteria want to communicate with red bacteria, it also need auto-inducer 2 machinery. Okay. In a system here full of different types of bacteria, we need to have both types of auto-inducing systems in a place. So in this case, we can look at this. So this is the molecule DPD, which is auto-inducer molecule, which will form the auto-inducer molecule. It can form its structure in such a way to make auto-inducer molecule with the LuxP ligand, with the LuxP molecule to make the auto-inducer 2 carrier, and it can also convert the structure in such a way to make LSRB ligand, which is also a part of auto-inducer 2 system, to make a different types of ligand. So in this case, for example, look in this case, in case of auto-inducer 2 system, red are Vibrio harvey, for example, and the blue are Salmonella typhimurium. Okay, so in these two types of pictures, what we are looking at in this case, in case of Vibrio harvey, they are making the instruction in such a way they produce the SDHMF and the THMF. Finally, it will attach the bromine residue here to finally make THMF borate, which will attach with the LuxP. This acts as the LuxP ligand, which will bind with the LuxP receptor molecules, and finally can be transferred from the outer space into the inner space of inner region of the cell. And this DPD can also be folded in such a way that they can make a molecule. That molecule can act as a signaling molecule for the type Salmonella. Okay, so in this case, the Vibrio harvey is making the DPD, and the DPD is when the Vibrio harvey needs to convey the message with another Vibrio harvey, it is making this THMF borate system, or this auto-inducer can be converted into THMF borate system. And when this Vibrio harvey wants to communicate with the Salmonella typhimurium, they needs to fold this DPD in such a way like this THMF way only, without any boron, and to make only one pentameric ring like that, to finally interact with the Salmonella typhimurium. So it's not the structure, there is a freedom of making the structure. So DPD is a generalized structure. We can fold DPD in such a way that they can interact with the Vibrio Harvey type of bacterial strain. You can fold the DPD in such a way they can interact with the Salmonella typhi or Escherichia coli like of bacteria through the system. So this is the modification scheme that helps the bacterial cells to control, to take a control over all these quorum sensing mechanisms. So these are some genes which are controlled by this autoinducer 2 molecule. So you can see, you can pause the video, you can see for different genes out there, and this type of bacteria along with these genes are as a topic of research in nowadays. So you can find the different research articles in the internet over with these different gene or gene products. So now let's come to the summary of it. So what is the quorum sensing in bacteria in few words? Bacteria talk to each other. This is the first thing, via the chemical signals or chemical messengers, like we call it the autoinducer. They have multiple languages. For example, there are multiple languages, like they have signaling, they have interspecies communication. What is the type of interspecies communication? With the help of autoinducer 1 pathway, where one bacteria communicate with the same bacteria of the same family. And what is the interspecies communication? This type of communication is carried out by autoinducer 2 pathway, where one type of bacteria triggers or creates some signal which helps to communicate with another bacteria of another different family. And bacteria distinguish self from other bacteria via this quorum sensing technique. That's a very, very important part of the quorum sensing. Okay. And many more molecules remain to be discovered, because this is the only 10 to 15 years of research, and finally you end up with something like that. So you generally do not find information in book, because this is really, really new, very new topic. So this is a very, very important region for active research in the future generations. So we need to find which molecule does what, and what is the necessity of the molecule throughout this quorum sensing system. And quorum sensing allows bacteria to be multicellular, similar to higher organisms. Why? Because in higher organisms they will communicate via different endocrine signaling or paracrine signaling and all different synaptic signaling like that. But in case of quorum sensing, the one bacteria can interact with another bacteria to finally make a colony, which we call a biofilm, with the help of these quorum sensing techniques. Okay. So quorum sensing is an active area of research now all the time, and we need to learn more about quorum sensing. Why? Because if we know about the quorum sensing, then we can make, or we can produce some drugs that will block the effectiveness of this quorum sensing, the effectiveness of communication of bacteria, intercommunication of bacteria. And when we can block this communication, the bacterial cells do not know whether they need to thrive any violence factor, we need to push any toxic material into the outside or not. So they will diminish, they can be killed by only knowing the mechanism of quorum sensing techniques. And as well as not only know the quorum sensing techniques via this, but we also by understanding the quorum sensing techniques, we can find some bacteria which are having the anti-quorum sensing strategies, because bacteria are ancient than us. Bacteria are really, really senior citizens. So they come into this planet many years ago than us. So there may be different bacteria cells or bacteria which will produce any different techniques to fight against the quorum sensing mechanism, to invade, to block the invasion of other types of pathogens. For example, here we have one pathogen, it will make a quorum sensing strategy, but this other pathogen, which is pathogen B, do not want this A to communicate with other pathogens, so it will block the action of this pathogen A. And we need to learn this kind of anti-quorum sensing strategies which already exist in the nature naturally. If we derive these techniques, if we derive those molecules which helps them to block this quorum sensing procedure, then we can block the formation of biofilms in regions like our tooth and all these places like that. Okay, so finally, so this is an active research area nowadays, and many years to go, many things to be discovered. So this is the open area of research, and that's the whole is the basic idea about the quorum sensing. This is a very minute area or summary of quorum sensing what I have discussed, but still it's going to help to establish the understanding of quorum sensing. and how the quorum sensing affect all these systems. Thank you for listening. I hope that's gonna help you.